Hello students, welcome to Academia. Today I will discuss about microtomy. So, many living things are extraordinarily beautiful, is not it? But some things are microscopic, we cannot see them. So to get a privileged view, this may be enhanced by looking inside. And it can be done by seeing under microscope. So if we want to see them under microscope, we have to cut them in proper slices and stain them properly to see under microscope. In this regard, for cutting the sections for the tissue preparation, because all these things are done under histology, so the tissue preparation steps, which include the fixation, dehydration, clearing, embedding, section cutting and staining. So in this section cutting process, which is the most important instrument, it is the microtome. Now what is microtomy? What is microtome? So microtomy is the process, the means by which the tissue can be sectioned and attached to a surface for further microscopic examination. The process is called microtomy and the instrument is called microtome. So where from the word microtome come? The Greek micros, which means small, and temnin, which means to cut. So from these words there is a word called microtome have come. So next we move to the history, that is what happened in the earliest ages. So then the freehand sectioning was done by using a sharp razor. Of the fresh and fixed material. Santiago Ramon y Cajal, who in 1884 to 1887, this scientist produced the microtome, whereas the Charles Sedgwick Minot, who invented the rotary microtome. So these are the broad classification of the types of microtome. The rotary microtome, rotary rocking microtome, base ledge microtome, sliding microtome, freezing microtome, vibrating microtome, saw microtome, cryostat, ultramicrotome and laser microtome. So these are the types of microtome, among which is the most important and mostly used microtome is the rotary microtome. And cryostat is a very specialized type of microtome where we use the frozen sections, whereas the ultramicrotome is used for the identification of the sections for ultramicroscopy. So these are the broad classification of microtome. So one by one we will see. Today in our discussion I will discuss about the rotary microtome, the parts of rotary microtome, the working principle of rotary microtome, the advantages of rotary microtome. Now we focus on the rotary microtome. So in the rotary microtome, which is the most commonly used microtome in all laboratories, basically the college or labs laboratory. So here there are three types. One is manual, which is used by using our hands or rotating the wheel. Another is semi-automated and fully automated. Okay. So in semi-automated, one motor to advance either the fine or coarse hand wheel, and in the fully automated, two motors that drive both the fine and the coarse advance hand wheel. So in the semi-automated, one motor is used, and in the fully automated, two motors are used. And in case of the manual rotary microtome, which we generally see in our labs, they are completely manipulated by the operator. Next, in our previous diagram, we have seen the parts of the microtome. Now, what are the major parts of the microtome? First of all, knife holder base. So in the parts there are the knife holder base. Next one is the knife holder. The knife holder. Then move on to the coarse hand wheel. The coarse head wheel. Then the micron adjustment. The micron adjustment. Advancement hand wheel. Safety lock. So these are the basic parts of the rotary microtome. Knife holder base, knife holder, then coarse hand wheel, micron adjustment, advancement hand wheel, and safety lock. And now one by one we try to understand then what are these things. So the first part, that is a knife holder base. So this is the part which anchors the knife holder, that means which holds the knife holder, and it can be moved toward and away from the block. The holder base can be moved, but it must be stationary and locked during the process. That is, when we cut through the microtome, it should be stationary and locked. Now the second part is the knife holder. So this part is comprised of some components like the blade clamp, then the knife tilt, and the face plate. Now what are the functions of all these things? So the blade clamp, as it revealed, that it holds the blade. So the blade clamp, what it done, it holds the blade. Then what about the knife tilt? It is used for adjusting the knife angle. So it is used for adjusting the knife angle. And the face plate, it guides that ribbons away from the blade and towards the operator. So what is the function of the face plate? So they guides that ribbons, that is the section ribbons, and towards the operator. So these are the three components with their functions. Next part is the coarse hand wheel, which are used manually, and it moves the block holder actually either toward the knife or away from the knife. So toward the knife or away from the knife. So the diagram below also shown that part. Next one is the micron adjustment, and the setting, we can adjust the setting, and it is the range of 1 to 60 microns on most of the microtomes. So we can set between these ranges 1 to 60 microns. So below that also there is the image of the micron adjustment. Move on to the advancement hand wheel, which helps in the turning in one direction and advances the block toward the knife at the specified microns. So it helps in the turning, and of course it should be turning in one direction, and it advances the block towards the knife at the specified micron. The last part is the safety lock. The safety lock are used to prevent the wheel which we rotate from releasing, and having the block holder come down toward the blade while a block is inserted or removed. So generally the lock is very essential when we are not doing the work. So it should be used anytime the microtomist is not actively sectioning. So when the microtomist blocks. So everybody who are dealing with the microtome, they should aware of the safety lock. After the working, they should lock the microtome. Otherwise, if the block is attached with the microtome, then there will be an easy cut, or the section or the block can be damaged. So the safety locks are very important part of the microtome. So these are in brief the different parts of the rotary microtome, and this rotary microtome is a very useful apparatus used in histology, basic histological section cutting. So what are the advantages of this rotary microtome? So there are many advantages of the rotary microtome. First of all is the ability to cut the very thin sections. Next, the heavier, and it is more stable. It is much heavy. The large, heavier knife also, which combat less vibration. The cutting angle of the knife is also adjustable. The easy sectioning of all types of tissues, whether it is hard tissue, whether it is fragile or fatty. Now hard tissues means the tissues which the composition is a bit compact. Then fragile means the tissues which can easily broken down, and the fatty tissues which have lots of fats inside. So all types of tissues can be cut by the rotary microtome, and the large number of sections we get from each tissue block. Lots of advantages are there, but also some disadvantages also there. It doesn't allow the serial cuts, which slows down the process. And one of the most important thing is that the exposure of the blade can cause the accidents. Generally nowadays we are not using the knives for our betterment. We use disposable blades, and if we don't take proper precaution, then there will be chances of cuts. So otherwise the rotary microtome is the ideal microtome using for the practical experiments of the histology specimens, and after that we can go for the staining process to see the different type of microscopic specimens. So this is all about the rotary microtome. If you have any query or question, you can ask box. Thank you so much.